welcome back. In the previous presentation, in part 1, we went through insulin's production and release, and now we will move on to part 2. In part 2, we will look at insulin's action specifically on muscle cells, adipocyte tissue or fat cells, and the liver. Alright, so here we'll just quickly go through insulin's action on target tissues. So the liver, the muscle, and the fat. So in the liver, it's going to increase lipogenesis, so lipid production. Next, it's also going to cause glycogenesis, so increase in glycogen production, which is the storage form of glucose, and also decrease gluconeogenesis, so decrease production of glucose. In the muscle, we're going to increase glycogenesis, so increase of glycogen production. It's going to increase amino acid uptake into the muscle cell, which in turn will help increase protein synthesis. And in the fat cell, it helps to increase fatty acid uptake into the fat cell, which helps to form triglycerides. And so this is basically increase in lipogenesis. So here we're going to look at insulin's action on the liver cell. So here we have a liver cell, and over here we have a blood vessel with glucose in it. So when we eat food, our blood glucose levels increase. And now the glucose can enter the liver. How is it going to do that? It's going to be going through the GLUT2 transporter. Again, this is an insulin-independent transporter. It doesn't rely on insulin for its mode of action, and it's more active when our blood glucose levels are high. So for example, after we eat food, so glucose will enter the cell and go down two pathways right here. Two pathways. One is glycogenesis, so it's going to be the formation of glycogen, storage form of glucose, and glucose is also going to go through glycolysis. So one of the end products of glycolysis is pyruvate. Again, pyruvate, we still have a lot of energy we want to extract from this. So it gets converted to acetyl-CoA. Acetyl-CoA goes through the Krebs cycle, which is again occurring in the mitochondria, and we end up with the end product of ATP, energy. Another side note is there's also production of fatty acids. Important side here is that when we eat food, especially food with a fat, there's fatty acids in the blood. These fatty acids will also make their way into the liver. So what's the end product here? Fat or triglycerides. And moving on. So here we have insulin in the blood. Insulin is going to act on its receptor, the insulin receptor. So let's briefly just go through the insulin receptor. It's a tyrosine kinase receptor. It has, so up here we have two alpha subunits, two beta subunits, and within the beta subunits here, there are the tyrosine kinase domains. And how this works is that when insulin binds to its receptor, this causes autophosphorylation of these tyrosine kinase domains, and this leads to a cascade of events leading to secondary messengers that are produced. And the main ones that we will just briefly talk about are the PIK3 and AKT, which is this protein kinase B. And these secondary messengers also have a role within the mechanisms that are occurring in the liver and also help with the formation of glycogen. And finally, just over here, we have just a summary of everything that's occurring within the liver. All right, so before moving forward, let's go through the GLUT transporters on the muscle cells and also the fat cells. Remember previously we went through the GLUT2 transporter, which was on the liver cell. So let's go through this for the muscle cell and fat cells. So here we have a cell membrane of a muscle or fat cell, and here we have insulin receptor. Remember, insulin receptor is a tyrosine kinase receptor. It has two alpha subunits, two beta subunits, and also disulfide bonds. Remember, within the beta subunit, there are tyrosine kinase domains. When insulin binds to its receptor, this will cause a cascade of events, and this will eventually lead to the translocation of vesicles which contain GLUT4 transporters. And these will be embedded into the cell membrane. And by doing this, this actually allows the glucose to enter into the cell. Just to re-emphasize again, the liver cell had the GLUT2 transporter, which did not rely on insulin for its mode of action. Now we see here with the muscle and fat cells, in order for the GLUT4 transporter to be embedded into the cell membrane, it required insulin to bind to the tyrosine receptor, which causes a cascade of events, eventually leading to the vesicle containing the GLUT4 transporters to dock in the cell membrane, eventually allowing glucose to enter into the cell. All right, so here we have just a summary of the GLUT transporters. Just to emphasize again, the GLUT2 transporters are primarily within the liver cell, the beta cells of the pancreas, and the small intestine. Again, these are insulin independent. They do not rely on insulin for their mode of action. They act more like sensors in that when our sugar levels are high, they are more active, which allows glucose to enter into the cell. On the other side, we have GLUT4 transporters. These are primarily in the skeletal muscle, the adipose tissue, or the fat cells, and cardiac muscle. And these are insulin dependent. They rely on insulin's mode of action. They rely on insulin to bind to its receptor in order for the GLUT4 transporter to be translocated into the cell membrane to allow glucose to enter into the cell. Okay, so let's look at the insulin action on the muscle cell. So here we have the muscle cell. Here we have a blood vessel with glucose in it, and we have a blood vessel with insulin. So in order for glucose to enter the cell, it needs a transporter. And how is that transporter going to get there? The first stage is insulin. It's going to act on this receptor, insulin receptor. Again, it's a tyrosine kinase receptor. It's going to lead to a cascade of events, eventually leading to translocation of GLUT transporters. Now, which one are these? Are they GLUT2 or GLUT4? They're GLUT4. And remember, GLUT4 transporters are insulin dependent. They depend on insulin for its mode of action. They depend on insulin to become to get translocated into the cell membrane. So now this has occurred. We have the GLUT transporter here. Glucose can now enter the cell. Again, glucose will go through glycolysis. We get the formation of pyruvate. We get converted into acetyl-CoA, and the acetyl-CoA will go through the Krebs cycle, and we will get the end product of ATP. And remember, with the muscle cell, it also stores glycogen, so glycogenesis will also occur. We've highlighted before that when insulin binds to its receptor, it leads to secondary messengers, PIK3, AKT, or protein kinase B. This will also aid in the mechanisms within the cell and also help with the glycogen production. And we're not done here yet. These secondary messengers will also affect another channel, and that is for specifically amino acids. So when we eat food from the meat that we eat, the amino acids from the blood will make their way into the cell. Amino acids, what do they form? They form peptides. And what do peptides form? Protein, which help to build muscle. All right, so let's look at the mode of action of insulin on a fat cell. So here we have a fat cell, and over here we have a blood vessel with glucose. And down here we have another blood vessel with insulin. So insulin is going to bind to the insulin receptor. And from here there's going to be a cascade of events, eventually leading to translocation of a GLUT transporter. And that GLUT transporter is GLUT4. And remember, GLUT4 is insulin dependent. So this is just like the muscle cell as well. And just to reemphasize insulin dependent, that it requires insulin for its mode of action. It requires insulin for it to be translocated to the cell membrane. And by doing so, this allows glucose to now enter the cell. And there's going to be a host of reactions now. So let's just go through one by one. So we're going to go through glycolysis. And with the glycolysis, there are many steps involved, and there are many products that are produced. So the two main ones here are the G3P, which is glyceraldehyde 3-phosphate, and DHAP, which is dihydroxyacetone phosphate. And you can see here there are arrows going back and forth, so they can be converted into either or, vice versa. The G3P will continue on to form pyruvate, and which will get converted into acetyl-CoA, which goes through the Krebs cycle. Again, it's occurring in the mitochondria, and we get energy in the form of ATP. On the side also, from acetyl-CoA, we'll also get fatty acids. Now, let's go back to the insulin receptor. So again, when insulin binds, there is a cascade of events, and we also have secondary messengers. So again, PIK3 and the AKT, protein kinase B, also it's called, and this is going to have an impact on another set of channels, and these are the fatty acid channel. So this is going to allow fatty acids. Again, this is from the food that we eat. So if we eat a fatty meal, there will be fatty acids in the blood, and this will allow the fatty acids to enter the cell. And now we have more fatty acids here. So from here, let's take a look back on these secondary messengers. These will also have an impact on this reaction here with DHAP and help in the formation of glycerol. When we take 3 fatty acids plus a glycerol, what does that make? It makes a triglyceride or fat. So we're also getting lipogenesis here as well. Alright, so that's the end of part 2, and that concludes the presentation. I hope you found this helpful, and again, thanks for watching.